Hello everyone. In the previous part, we learned about GERN system. In that, Golgi complex, endoplasmic reticulum, and lysosomes, how they are uh, interconnected and how they are working is learned in the last class. Today, we learn about another cell organelle, that is ribosome. Ribosomes. Ribosomes. These ribosomes are first observed by Robinson and Brown. Ribosomes are observed by Robinson and Brown in the root cells of Vicia faba. They observed in the root cells of Vicia faba. This is a plant. In this plant, the roots are collected. In the root cells, they observe ribosomes. And these ribosomes are isolated. By clade from cytoplasm. He isolated ribosomes from cytoplasm, and he named as microsomes. Later, these microsomes are named as ribosomes by Palade named these microsomes, or otherwise the term ribosome is given by Palade. That's why these ribosomes are also called Palade's particles. Ribosomes are generally non-membranous structures. They are non-membranous structures. Membrane is absent. That's why they are called non-membranous structures. These ribosomes are found in eukaryotic cells as well as prokaryotic cells. Ribosomes are found in eukaryotic cells as well as prokaryotes. Prokaryotic cells, prokaryotes like bacteria. In the bacteria cell, ribosomes are present. Like that, in the eukaryotes also ribosomes are found in eukaryotes. But there are some exceptions. Exception means in the eukaryotic cells, in some cells, ribosomes are absent. There are some exceptions. What are those exceptions? They are absent in mature sperm cell. Mature sperms. And they are also absent in mature mammalian RBC. Mature mammalian RBCs. These are the some exceptions in which ribosomes are absent. These ribosomes are also found in cell organelles. They are also found in cell organelles. Cell organelles like mitochondria and chloroplast. If the ribosome is present in mitochondria, they are called mitoribosomes. If they are present in chloroplast, they are called plastidoribosomes. Mostly ribosomes are present in the cytoplasm of the cell. They are generally present in cytoplasm of the cell. Cytoplasm of cell. Those ribosomes are called free ribosomes. Free ribosomes. Free ribosomes. If the ribosomes are attached with membrane, then they are called bounded ribosomes. For example, endoplasmic reticulum is present. On the surface of the endoplasmic reticulum, ribosomes are present. When they are attached to membrane, then they are called bounded ribosomes or bound ribosomes. Bound ribosomes. These ribosomes are attached with the membrane by a special type of protein, that is ribofourin. They are attached to the membrane by a protein that is called ribofourin. With the help of this protein or enzyme which attaches to the surface of the endoplasmic reticulum, otherwise nucleus. Okay. Next, ribosomes have two subunits. This is a ribosome. Ribosome have two subunits. They are larger subunit and second one is smaller subunit. Smaller subunit. For example, this is a membrane. This is a membrane. With the membrane, with the help of ribofourin, larger subunit is attached, and on the larger subunit, smaller subunit is present. Ribosomes have two subunits, larger subunit and smaller subunit. Larger subunit is larger in size and which is attached to the membrane, with the help of the enzyme ribofourin. And smaller subunits are generally free. When the protein synthesis takes place, then these smaller subunits are come into contact with larger subunits. And again, ribosomes are divided into two types. These ribosomes are divided into two types based on their coefficient, Swedberg coefficient. They are 70S type ribosomes and 80S type ribosomes. 70S and 80S ribosomes. 70S type of ribosomes are found in prokaryotes, prokaryotic cell like bacteria, and 80S ribosomes are found in eukaryotes, eukaryotic cells. They are found in eukaryotic cells, and 70S type ribosomes are found in prokaryotic cell. Here S stands for Swedberg units. Swedberg units. S stands for Swedberg units. Otherwise, S is also stands for sedimentation coefficient. Sedimentation coefficient. That means when the cytoplasmic content are filtered, these ribosomes are settled at 70 sedimentation coefficient. That's why it is called 70S type of ribosomes and 80S type of ribosomes. Again, this 70S ribosome have two subunits, larger subunit and smaller subunit. Larger and smaller subunit. Larger subunit is about 50S, and smaller subunit is about 30S. They are the Swedberg units or sedimentation coefficient. Next, coming to eukaryotic cell, 80S type of ribosomes are present. In that also two subunits are present. They are 60s is the larger subunit and 40s is smaller subunit. 40s is smaller subunit. This is about small introduction of ribosomes. In the next class, I will explain different types of ribosomes and their functions. Okay, thank you.